good to go.
Good afternoon from what is a very warm UK at the moment. This is the first session on the first day at the Future of Biopharmaceutical Analysis Summit 2026, looking at modernising environmental monitoring. And we've got two fantastic experts today. We have Michael J. Miller, President, Microbiology Consultants, and Kate Coleman, Vice President of Contamination Control, Microbial Excellence and Analytical Services at Thermo Fisher Pharma Services Group. It's going to be a fantastic session, probably one of the best that we've got. So I'm going to hand straight over to Kate, and she's going to go into the first slide.
So welcome everybody. I'm here with Michael and we're going to have a discussion today on our experiences with environmental monitoring and where it is in the industry today, and the methods that we have that we could try and modernize the environmental monitoring, particularly in pharmaceutical manufacturing, as it gets more innovative and we get more technology moving through the industry. So really, environmental monitoring is a cornerstone of contamination detection and regulatory compliance in pharmaceutical manufacturing. It is the one detection method that we really rely on to tell us that everything is working well from a contamination control point of view. It's a key detection in our contamination control strategies. And as product innovation is accelerating, with emerging modalities, advanced therapies, ATMPs, CGTs, really increasing contamination control complexity, environmental monitoring has relatively stayed the same. The traditional methods really remain the benchmark. And even with the emerging modalities and advanced therapies, increasing that complexity, the methods that we use for environmental monitoring, they haven't really kept pace with the other innovations within the industry. And this is coupled with the fact that at the moment in the industry, real-time insights are increasingly required. We want to have real-time signals, particularly in isolators and more modern technologies, of where are we from a contamination control point of view, so that we can take those signals, take action in real time, make sure that we're protecting the product. But the current traditional methods do have delayed results and they do limit that proactive decision making. Future programs, they really need to be designed to be predictive, data-driven, able to get the data out of the system quickly. And as I said, to make sure that we're reacting to those signals. But the integration of those advanced technologies will enable a proactive approach. But, how do we go about that? So, Michael, I don't know if you have a thought on why we're still at this point where we have the traditional methods in environmental monitoring, even though the industry is moving on so quickly with technology.
Right. And so good morning, Kate, and good afternoon, everyone else. Listen, the standard for environmental monitoring hasn't changed in decades, right? And sometimes change is a little difficult. It's a little scary. And although we have a handful of companies that have moved toward more rapid, automated, alternative microbiological methods for environmental monitoring, the majority of the industry is still locked in with the manual, standard CFU-based environmental monitoring, because it does take a bit of work to evaluate and put these systems in place. That doesn't mean we don't have the framework for being able to do so and demonstrate comparability or equivalence, because we do. We do have the guidance documents to do that. We have the regulatory acceptance for these new technologies. We're just needing to get over the hump, if you will, and help promote not only the need now with these new modalities, but it is much easier today than it was in the past to be able to validate and implement these methods going forward. And Kate, I know we're going to have some good discussions as we go through today on how do we actually make that happen.
Yeah, and if we move on to the next slide, Ian, I think that will bring us to, the comparison between the traditional versus the rapid and the advanced EM methods. So, as we've mentioned already, industry is really moving from that retrospective detection to a predictive contamination control. The requirement is clearly there to move to a predictive, system and process for detecting contamination within our processes. But, as we're talking about, why are the traditional methods still the gold standard? Where there's regulatory acceptance. We know that it is an accepted standard. We have historical data sets. A lot of us in the industry have been, looking at our trends for years, and we can see those signals over time. Although they're not the most predictive in terms of looking forwards. There's established standards, there's the USP, there's Annex 1, ISO standards, and they all talk to those traditional methods. There's strong comparability as well. You can compare one plate to another in the same location and get an estimation of, what your performance is. And a big one, I think, for the industry as we move very quickly is lower cost and validation burden. We know how to validate these methods. They're relatively simple, but there are challenges. So there is a lack of sensitivity. We all know that although environmental monitoring is a good indicator, it's not the most sensitive method. And it is that it's an indicator, whereas with some of the other newer innovative methods, there are, abilities to see something in real time that's a little bit more accurate, which also brings, how do you manage that with your action limits, etc. It's not really suitable for predictive real-time action. The traditional methods is a distance from the operator interface. We're not giving people real-time feedback. You don't know straight away that a settle plate has had a hit until 7 days later, sometimes 8 or 9, depending on what way your process works. And there is a manual workload. They're physically putting them in. Although that has changed and there are robotic solutions out there, it's still relatively manual. And then regulatory expectations. So the advanced methods, faster detection of excursions, near real-time decision-making and feedback to production are some of the advantages. Reduced incubation delays. We all know those challenges when there's a critical part of the process and you want to know before 7 days that everything is okay. Your improved contamination control strategy effectiveness, again, because you've got that real-time data and enhanced data integrity and visualization. But maybe Michael, I'll leave you to talk to some of the challenges, because I know you've got a lot of experience in this area as well, in terms of how to validate these newer methods. So maybe if you want to go through some of the challenges and maybe how we move past them. And I think that will lead us into the next slide as well, Ian.
Yes. So thank you, Kate. Listen, I've been working with rapid and alternative methods for almost 40 years. And we can go back to that prior slide for a moment, because I wanted to address some of the bullet points on the challenges below. And I think the number one here, if you ask stakeholders why they're not moving forward with the evaluation of an alternative method, even for environmental monitoring, two big things come up. One is the lack of validation guidance, which I know we will get to in the next slide, and I will debunk that misconception. But more importantly, it is the regulatory landscape, and there are many stakeholders in the industry who believe that the regulators don't understand alternative or rapid methods and don't accept them, and that's just not true. All we have to do is take a look at Annex 1. When you read Annex 1, the revision to Annex 1, it is very clear that the EU is moving the industry away from the traditional environmental monitoring and encouraging us to go toward the next generation of EM. Here is a very specific example. There are two tables for classification of controlled environments and the routine monitoring. And right now, these tables do have what are considered to be acceptance criteria for CFU-based systems. But there are footnotes in these tables, and the footnotes make it very clear that you can utilize alternative methods, including rapid methods, and the term rapid methods we can find in the main text. And that if you go to one of these other systems, and we have demonstrated that they are equivalent to the existing CFU-based methods, that we may be able to change those numerical values to be based on the new rapid method signal. Okay, so this is the first time that we have a true regulatory document that is encouraging us to use these new technologies, and we have the option of showing a correlation to the existing CFU method, and even moving forward to making some new acceptance criteria based on those new technologies. The microbial identification challenges piece, that has always been a challenge, because whenever we get a hit, there is an expectation that we try to understand what the organism is so that we can ask the question, have we seen this organism before? Is it coming from the same location? Where could the source be? And some of the newer technologies that have been introduced, such as biofluorescent particle counters, historically, and I worked with the first one back in 2007, did not have the capability of truly capturing the airstream and what the particles might be, including viable organisms. We're starting to move away from that with the introduction of some workflows in which we're trying to capture those organisms. And then there are other technologies, not necessarily continuous, but there are now technologies based on AI native biophotonics where you can get a detection and a quantitative count, but also on microbial identification. So the science is now moving in the direction that we always would like it to be. There's still some evolution around this, but we certainly are moving forward. So industry, don't be scared because we do have the framework. And I think, Kate, as we go to the next slide, we'll be able to dive a little deeper into this.
Yeah, and exactly. And as we go to the next slide, I think it really is time that we start looking at how we can actually implement these methods, because even speaking to the regulators, if we can show that we can get those predictive signals and actually take action before something becomes, an issue, or that we get that data and we can take action in real time while the product is there on the line, that's always going to be preferable as long as we have the scientific rationale. And I know some industry operations people will also be worried about, the space that they have within their equipment. Isolators are restricted, and how do we do that comparability study? But I think a lot of these things, there's now starting to be precedents for how they can be done and how they can be managed. So in terms of implementation then of these rapid EM methods, there is a validation framework, very high level. But first of all, we need to define the intended use and the risk assessment. So it's looking at things like, what are we expecting this methodology to do? The ID question will come in here. Are we expecting it to be something that gives us a real-time count, or is it something that we do expect to be able to recover a sample and do an ID, and it fully replaces our traditional EM? But we need to define what is it that we want it to do. The big one is the demonstration of equivalence to the current method. And that's always the first hurdle, I think, to get over, because once you've done it once, and I think of manufacturers, like ours, where you've got multiple isolators, you might have a lot of the similar type of equipment. And once you've done it once, that's where you get over that big hurdle. But doing it once, that is the tricky part. And then looking at accuracy, precision, robustness, repeatability, your limit of detection, your recovery studies, all of that needs to be covered, because again, you want to make sure that the results that you're getting, that they're going to be more sensitive, but you want them to be accurate. You don't want to be reacting to so many signals that, it's difficult to get through your process. So you need to look at each of those items and make sure that you've got, I suppose, ranges around them and what you're trying to achieve. On 4, then it's correlation across your representative microorganisms, making sure that there's nothing that you're, leaving out of the ability to detect from your process. So again, it's similar to any other traditional EM method. You need to show that your representative microorganisms are going to be detected through that method. And 5 is really there establishing your alert and action limits. I think that's another key one that people, are nervous of how do we actually set those alert and actions? How do we show that they're equivalent to the traditional methods? And how do we make sure that the signals we're getting are true signals? And that feeds into your trending strategy as well, which if you're introducing these methods, you want a trending strategy that's a little bit more real time than what we maybe have done traditionally. And as you mentioned, Michael, talk about the identification strategy, although I think, and both of us have had this experience of seeing this, it has moved on. So, if you look at 10, 15 years ago, these types of methods, you really didn't have an opportunity to ID. But I think the industry has listened to that, and there are strategies there now that work quite well with these methods. So your
Right.
challenges again, and I'll hand over to you, Michael, here, so you can give some of your perspective in terms of the experience with validating these. But I don't know if there's anything you want to add to the validation framework as well.
Yeah, so if we look at number 2, which you've really highlighted, demonstrating equivalence, or what we prefer, the term we prefer to use today is comparability, or
Yeah.
non-inferiority to the existing method. The question is, where do I find a roadmap how to actually perform this. How do I know what the validation criteria are and what the acceptance criteria should be to be able to demonstrate comparability? And I want to point our listeners to the PDA, Federal Drug Association Technical Report number 33, which since 2000 has been considered to be the ultimate guidance on how to validate alternative and rapid methods and how to implement them and compare them to the existing methods to a point to where the regulators would accept that data as demonstrating comparability to where you can move to the rapid method and go completely away from the traditional method. The first version came out in 2000. It was revised in 2013. And I was the chair for that process. And since 2013, there have been new technologies introduced, new regulatory expectations. Of course, Annex 1 came out. And so for the last 3 years, I have been co-chairing another revision to TR 33 that answers a lot of the challenges that, Kate, you have put up on this particular slide. And believe it or not, after 3 years, we have the final approval, and we should have the TR published in the next couple of weeks, so everyone can watch out for that online. Most importantly is the question of these next generation alternative methods, which will present data which is not the colony forming unit, the CFU, but is presented as some other signal, like an autofluorescent unit or something similar. And of course, the question is, how do I compare an AFU to a CFU when I know there are organisms that may never grow on an agar plate? If we look at stressed organisms, if they are present, or as you say on the slide, viable but non-culturable. And if I have continuously monitoring EM systems, how can I compare that to a single point, which we normally get with a traditional method? So the technical report has specifically added a section discussing the validation options that stakeholders can have for comparing a non-CFU signal to a CFU signal. For example, if we can do it numerically, that's fantastic. If it's not possible to do that numerically, then we may be looking more at trends or the presence of non-zeros, or equating what would be the alternative method signal result to when we would normally expect the traditional result to give us a failing result, okay? And so there is now going to be, for the first time, new guidance on how to work with these non-CFU alternative methods and how to compare them to the existing method. And of course, there's going to have to be some side-by-side testing to generate the data, but we're not talking about a huge PhD project here. You'll have the guidance on how to do that. Part of that validation should include an assessment of whether false positives, as you have in your bullet list, false positives from non-microbial fluorescence, whether or not you're going to get an EM hit with these new technologies. And I will tell you, when I first started working with a biofluorescent particle counter back in 2007, one of the first things we did was we were running the system, and at the same time, we were using 70% IPA within the isolator. And the IPA got sucked into the instrument, and not only did it ruin the optics, but it also gave a flurry of false positives. Okay, this was a learning experience at the very beginning. But as part of your validation in-house with these systems, this is something that you will want to do, and the suppliers can help with this type of assessment, is to determine, are there any non-viable particles or cleaning agents or residual VHP or anything else that might elicit a false positive response, and then you set up a strategy for clearing that area before you routinely run the environmental monitoring. So of course, false positives are going to be looked at. As long as you generate the appropriate data to show through method suitability studies and compatibility in the environment and some side-by-side testing, the regulators are going to be in a much better position to understand the testing that you performed and put them in a position of accepting your scientific principles on demonstrating comparability. The one thing before we leave this slide I do want to say is certainly a challenge is many companies have tried to take these new methods and do side-by-side testing in a grade A environment. Okay, now folks, in a well-controlled grade A environment, we know we get zeros on the conventional method, but we would also expect zeros even with the rapid methods, with a non-CFU based method. You cannot compare zeros to zeros, all right? And so what the expectation here is, although you can show that you're getting some zeros with the alternative method in your grade A environment, to demonstrate statistically that the system can detect microorganisms and do so non-inferiorly to the existing method, it is appropriate to do some additional side-by-side testing in less controlled environments. So here, a lot of companies have gone to grade C and grade D areas to demonstrate that whenever you do get hits in the conventional method, you are certainly getting hits in the rapid method as well. And again, if you go back to the new TR33 that will come out very shortly, we do provide guidance on how to actually perform these types of tests. So a lot of information, but it was important to go through to make sure that all of the listeners understand this is not an insurmountable task, but you do need to go through certain steps in order to put these systems in place.
I think it's a good time as well, Michael, to link this back to the contamination control strategy, because particularly to your point there, where we speak about grade A environments and the move towards isolators. And we all know that, they're not perfect. We have to do a certain amount of pre-VHP setup. We need our material transfer to be correct. We need our VHP to be reliable. So all of those things that layer together iteratively as part of your controls within that isolator, they really do need to be managed very, very well when you're using a rapid method, to make sure that you are getting those zeros in your rapid method like you get in your traditional methods as well. Because I think we rely sometimes on those traditional methods, and when you do have maybe a signal that you wouldn't have had with a traditional method, there is maybe a nervousness about that. But it's really about implementing that contamination control strategy, those controls, and making sure that your environment within that isolator is truly managed the way that it needs to be. And I know that would be something that will cause a little bit of nervousness. And the other one that I'd like to just call out as well is, where we do get a genuine hit, you are now going to be I suppose, getting signals for bacteria, non-culturable microorganisms that you may never be able to ID, because to ID them you may need to culture them. So, there's always going to have to be that ability now to learn how to manage hits that we do get without knowing that ID, and just taking it for what it is that you've got contamination in your environment. So there's a couple of different challenges there to think about in the broader contamination control strategy as well.
Yeah, Kate, you bring up a very good point, and this is another sticking point with some in the industry, to think that if you put one of these new systems in place and all of the other controls are working, right? You have a validated and robust VHP cycle of whatever other gaseous material that you're using. Your HEPAs are working properly, your airflow, your changes, and so forth. Your gloves are integral if they have them, or you've even installed a robotic system for manufacturing within an isolator. These are not sterile environments, okay? We cannot say 100% that there may never be one organism that gets in, even if it's viable but non-culturable or stressed. And so if there's a company that believes that if you're running in this type of system, when all your controls are working and you get a single viable hit in a rapid method that is not based on a CFU, this does not mean all of a sudden you're out of control, especially if we're running continuously, continuously for 8 hours, continuously for an entire week during a campaign, which some companies certainly do. I wouldn't lose any sleep over a single hit. What I would lose sleep over is if I see and out of trend finding based on the historical testing that I've done with that same rapid method. If I see a hit and then a few minutes later I see two hits and then it continues, that tells me, hey, now in real time, I see I'm starting to go out of control. I have an out of trend type of a finding, and that's when you can stop everything and do your investigation. And the interesting thing here, and I wrote a return on investment paper about this using a biofluorescent particle counter, everything that has been filled and stoppered and capped and is out of that manufacturing isolator downstream of when the event started happening, all that product is okay.
Yeah.
All of the product in bulk upstream before it entered that filling environment is still okay. So we can deal with in real time an investigation as to why we're starting to see that out of trend finding. And once we resolve that, we can do a standard line clearance and start up again, knowing that the environment is it back in control because we have a real-time system to tell us that that's the case. And then you continue manufacturing. So instead of getting a hit and then you're throwing out a huge medically needed biologics batch, we don't have to do that anymore, right? So there's
Exactly.
a lot to consider.
Exactly. And this is where then it starts making sense, where we've seen the move in the industry towards, making sure we're using sterile tools when we're doing interventions, not using the actual gloves, which, 10 years ago would have been used, not breaching first air, having everything in first air sterile. So as we move towards maybe using rapid EM methods more and having the possibility of that one signal, all of these things actually start to make sense, which this is kind of the last piece of the puzzle of where the industry has been moving. So maybe we'll move on to the next slide and look at kind of where we go from here. So, really what we're trying to move towards, and that's why I bring up the contamination control strategy. I bring up, some of the things in Annex 1, for example, that have really become hot topics and important since 2022. It's all leading us towards this advanced compliance model. So where it's real-time rapid EM really sits within that model, where we can be, moving from reactive to proactive contamination control using real-time data. I think you gave a great example there, Michael, of how we would actually manage if you had a hit in real time. And as you said, you're going to have the ability to know that everything before and after that point is okay. You've got probably a bigger data set now as well, so that you can actually have that view of the entire batch, which then helps you not have to reject everything because you've got one hit. You're going to have your EM and your HVAC, your personnel, your cleaning and process data all integrated and hopefully automated systems. There's AI driven solutions there that can also help with looking at that data and managing it again in real time, automate escalation and investigation workflows. All of this can go into a data lake that then helps some of your other automated systems in your QMS, for example. Risk-based sampling is something we have already, but this is when risk-based sampling comes into its own. So if you've got your locations for your rapid EM in the, where there's the highest chance of contamination, then it's the same principle as what we would have used for traditional methods. But I think I would argue they actually become even more powerful, and your risk assessment becomes more powerful when it's done correctly. Continuous monitoring then in your critical zones, real-time dashboards and alerts. So you can see a little bit like, any control center, and you see what's going on in real time. This is where your environmental monitoring, the contamination control, it all becomes much more front and center in terms of monitoring your batch, rather than something that we look at afterwards, when everybody's, cleaned out the isolator or the RABs and gone home and the EM's in the incubator. Predictive trend analysis, where are we moving towards, and then preventative, using that data to prevent the deviations. And there's a lot of discussion in the industry, obviously, around deviations and deviation numbers, human error, all of the rest of it. This really should be something that you can take action before it becomes an issue that you then have to investigate reactively looking backwards. So I think we all want to move towards this advanced compliance model. It sounds great in practice, but how far do you think we are from that, Michael, from your experience?
Yeah, listen, we're already there. Okay, it's just a question of how far do we want
Adoption.
to adopt this? Right. How far do we have
Yeah,
to adopt it? And I can tell you there are a lot of stakeholders in the industry who have already adopted this. Even if they haven't put the rapid methods in place, there are components of what we see on this slide that has already been adopted. And it's interesting. I want to point back to the 2004 FDA guidance document on process analytical technology, PAT. We're starting to do microbiology as PAT,
yeah,
but this is also aligned with what is in the revised Annex 1. And I just want to, read for our listeners what is in Annex 1. It's very clear that processes, equipment, facilities, and manufacturing activities should be managed in accordance with QRM principles to provide a proactive, not a reactive,
exactly.
but a proactive means of identifying, scientifically evaluating, and controlling potential risks to quality. This is where we need to bring the industry to. And I know there are stakeholders who are actually going down this path, but now we are taking it even further.
Yeah. And I think it'll be very positive for the industry as a whole, because being able to react in real time is really, I suppose, what we've been missing over the history of the last 40 years. Being able to know what's happening in real time really gives us that opportunity to push the manufacturing forward, and while also maintaining, patient and product safety. So maybe moving
Right.
on,
Yeah.
Ian, to the next slide. So the key takeaways then, I think, for our audience, and I see there's a couple of questions coming in that we'll move to in a moment, but really it's, traditional methods, they do remain the regulatory benchmark, but that doesn't mean they have to stay that way. As long as we are utilizing our data sets to provide that scientific rationale and, to be able to show that these methods are superior to what we're currently doing in terms of getting us to that advanced compliance model, that will move eventually, and we will move away from those traditional methods. The advanced technologies do improve speed and situational awareness. I certainly, in working with the teams we work with, look forward to the day where, you're doing something on the line and somebody gets that real-time signal and goes, oh, okay, we need to stop, we need to fix, and then, we can move on without actually impacting what are very, important therapies. And we don't want to be wasting product if we don't have to. Validation, then, I think we probably both agree it's maybe the thought of the validation is the primary barrier to the widespread replacement. We've done validation in other innovations in the industry, and they've been done very successfully. You mentioned, Michael, we've got robotic isolators now that don't have gloves, and they're able to manage environmental monitoring. We've really moved on in terms of what we're doing, and we've validated those types of technology. So there's no reason why we can't do that with EM as well. Hybrid programs at the moment probably deliver the best near-term compliance strategy, but they should be moving towards that more predictive and proactive solution. And future EM programs, they're going to be increasingly predictive and data-driven. And who doesn't love data and being able to make decisions based on data, and takes all that subjectivity out of it. I'm certainly looking forward to it. So I don't, Michael, if there's any other key takeaways you want to share with the listeners.
This is spot on. I think we've talked about a lot to now encourage our attendees to ask additional questions if they do have some. We may already have some questions that have come in to continue our conversations. But at the end of the day, the industry needs to make a decision. Do they want to continue working in the 19th century world of microbiology, or do they want to move forward and work with the new technologies that have been developed in the hopes of continuously improving our product quality in the grand scheme of public health and patient safety? That's the decision that companies within the industry need to make. And now we have the tools to be able to get to that point.
So I think it's probably time to hand it over to the audience and any questions that you would like to ask. So I think, Ian, are you keeping an eye on those?
I certainly am, absolutely.
Great.
We have got time for additional questions, so please put them in the chat if you'd like to. The first question that's come in is, and I think Michael already one-word answered this, so it'd be good to get both of your perspectives. You mentioned that it's ideal to do equivalency studies in grade C environments. Would you consider this type of system useful for standard use in BCD, where there is not a expectation of sterility or zero CFU?
Oh, absolutely. Actually, some of the earliest adopters for these biofluorescent particle counters implemented these systems in grade C and D environments. And the reason I said that it would be appropriate to do the side-by-side testing, and again, refer back to the technical report 33 for specific guidance on how to do this, is that when you're generating numerical data in terms of recovery of microorganisms, if you want to perform a statistical analysis to show non-inferiority or comparability, you need numbers, right? That's why I said you cannot statistically compare a 0 to a 0. You have to consider doing something else. But if you have numerical values in a grade C and D, where we expect to see some recovery, and still expect it to be within the acceptable levels for those areas, with those numbers, now we can statistically compare and demonstrate that the alternative method is not statistically worse at recovering those organisms. So in a grade C and D area where we generate actual data, it's perfect for the initial validation, but you can continue to implement those systems in those same areas for routine use. Again, if we go back to Annex 1 and we do happen to find that there was a greater recovery in those environments for the reasons we've already talked about, physiological stress, viable but non-culturables, as long as you have the scientific data to justify this, Annex 1 is allowing you to make modifications to those acceptance criteria. But of course, those new methods need to be qualified as being comparable first. You can't just plug and play and do this. You do have to do some work to scientifically justify the change.
And I think it makes perfect sense to be using them in grade C and D. If you think about, the every day of an environmental monitoring analyst, when they go up to the area and they do their environmental monitoring, they want to be able to tell people that day that you have an issue and that you need to do a clean and that you need to take action rather than waiting for 7 days. And you may have, a couple of batches actually made. It could be the background to an isolator, your grade C room, and maybe you've made a couple of batches in those few days. So it actually makes perfect sense to have that predictive data so that you can take the action at the time. And I'm certainly aware of these methods being used, including in investigational circumstances where maybe there has been hits on traditional methods. There's not enough data to see where it's coming from or to see the trend, or if there's certain parts of the area that are higher than others. So these portable types of equipment as well are also used for investigational purposes, for example, already in the industry. So there's definitely a precedence for them being used even more for C and D. I think where the comparability piece, although it's not statistical, there's probably, whether it's right or wrong, an expectation that you do run them side by side, at least in a grade A area, just to show that you're not going to have problems with, as false positives. But I think that's more of a comfort factor in the industry than a true necessity, to your point, Michael, in terms of you're better using data, like we would with any other EM method and a traditional EM method. But there is a comfort factor there, I think.
So our next question is quite a specific one. What technologies exist to replace the RODAC plate-based
RODAC
surface? RODAC. I was betting that was an acronym. RODAC plate-based surface sampling method.
Yes. So the answer is yes. And we can we can replace RODAC because this is surface sampling, and we can expand this to swab surface sampling for what would be considered flat or irregular surfaces. And without being biased, I've had an opportunity, instead of retiring, I've had an opportunity to work with a Europe-based startup who developed a technology that fits right into this, and it's actually being used as a replacement for Rodax and swabs. And it is based on transformer based multimodal spectral imaging, which is biophotonics and AI driven to generate the data, where you can take those surface samples and then put them through the particular system, and close to real time, you are getting a detection or a quantitative value, but also a microbial identification at the same time. And so there are other technologies as well that can fit into this particular paradigm. Some technologies that have been around for some time that offer very low detection and quantitation, not necessarily the ID piece, but certainly the quantitative piece, like solid phase cytometry or ATP bioluminescence and things of this nature. So we do have technologies available today that can take that Rodak plate, which you still have to put into an incubator, or a traditional swab where you still have to put it into traditional media or run it onto another agar plate. Instead of waiting the days for that, we now have these new next generation technologies that can give us results in close to real time. And when I say close to real time, we're talking about minutes. And again, now we can be in a better position to act upon excursions if we do see them on these particular types of surface samples. So that was a long answer, but I wanted to provide some scientific rationale and some background so that the listeners can go out and look for these new systems and see where they can make an alternative change to the existing Rodak plate and surface sampling.
And it really is a move on as well that's kind of happening in the industry anyway, when you again look at the likes of isolators, where it's really not appropriate to be using Rodax, even if it's a flat surface, because of your agar residues. And we've moved on in terms of even the type of sampling we do in those locations. So this next generation is going to bring us even further along that journey, where you're going to get a result without actually impacting, the quality of the surface. So I think this is going to be a very obvious next step to adopt.
So the next question is hybrid systems. Would this be where rapid methods would be the routine monitoring, but traditional methods for ID where hits are being found, or how would it work?
Very good.
I've seen it both ways. So and Michael, you can lend your experience here. So, but I've seen it both ways. So I've seen where the rapid method is implemented, and as the person who asked the question said, traditional methods as a backup for ID. But I've also seen the traditional method been primary, and the innovative method been kind of put there side by side to see what happens and see could we do comparability. And that can sometimes cause difficulties in terms of, the actual space that you have, let's say within a RABS or within an isolator. It can, cause some difficulties. But I think this is where really looking at what you want to do, what you're trying to achieve, why you're implementing the system, doing that risk assessment, having the comparability from maybe your CD areas first before you start putting it into an isolator. This is where it's really important to plan out what you're doing. And I'm not sure if you agree with that, Michael.
Yeah, no, I do. But we have to separate out using a hybrid system, both methods during qualification, which we would expect to do, versus using a hybrid system for routine, because that's a double-edged sword. What happens if one system gives you 0 and another system gives you a recovery? The regulatory authorities are going to look at that system that gave you a recovery, and they're going to be asking some pointed questions, right?
I think that's the challenge in an isolator. Yeah, it's a big challenge. Yeah.
So we have to be careful in justifying why we would use both of the methods at the same time for routine monitoring. Now, the comment, or rather the question, link this also to an ID, because there are some rapid methods that may not have the capability of giving an ID or even capturing microorganisms. And I'll be very honest, depending on the physiological state of the organism, some of the newer rapid methods that try to capture organisms, you may capture it, but it may not grow anyway, right? And so a hybrid system might include the following, which is what I recommended probably 15 years ago when these systems first came out, is if you're using a rapid method and you start to see a hit, not a single hit one time in 12 hours, but if you start to see a trend, you know there is a contamination event going on or a loss of control. That may be the best time for you to take a conventional method and try to capture those organisms to grow them for subsequent investigations, including microbial ID. But of course, for all the reasons we've discussed, Kate and Ian, sometimes the microorganisms might be there, but they are not going to grow on a traditional agar-based method. But it's better than not doing anything, right? So
And
there's a give and
yeah.
take here.
And I think specifically for the isolator use case, it's something I'm sure some of our listeners are thinking about, because isolators do get spec'd sometimes with both methods. So they get spec'd with, your air samplers in fixed locations, your non-viable isokinetic heads in fixed locations. And then they also have locations for the rapid method. So the question then becomes, once you've done that initial comparability, you still have all of the, hardware there. So what do you do then to roll back? And that's the piece about having it very well documented, what your intent is up front, what your design intent is, what your intent is after, you've gotten that initial data and comparability, and then how are you going to move away maybe from that traditional method, and how are you going to use it as a supplement. So I think a lot of the time you need to think in advance of what your long-term plan is, because what we sometimes see is that, the equipment goes in and then, you get your data and okay, what do we do now? We still have our traditional isokinetic heads, we still have our traditional air samplers. So that's where really thinking ahead of what your longer term plan is really important. And having that documented as a glide path to where you want to go.
I think we never should underestimate the necessity of documentation. I think
Yes.
that's something that often we do forget. Next question. So, is it required to take sampling of active and passive from the same point of location? Can I use both technologies in different points of the same room or area?
Kate, I'm
So
going to let you start with this because
yeah,
I've got my own personal views about active and passive sampling in the same location. But I'll let you take a
my
crack at this.
well, my view is there are different types of monitoring required for different things. And you have to, when you're doing your environmental monitoring risk assessment, one of the things, in my history of doing these is always looking at what it is you're trying to capture. How do you think, that contamination is going to manifest? Is it going to be something that does spill over, let's say, a stopper bowl? Or is it something that is actively going to be in your air? Is it something that's going to be transferred onto a surface? So I don't think they necessarily need to be in the same place. I think there needs to be an appropriate type of monitoring for the appropriate contamination source. That should be understood really well in your risk assessment, because your risk assessment is supposed to ask, what is my contamination source? What's that vector for contamination? And then you have the appropriate type of sampling for that. So it's not I need an air sample, therefore I also need passive. You may not need that if there isn't, an airflow going over. The stopper bowl is always the one I suppose I think of, where your air comes over and you do potentially have fallout. But you have to think about what the pluses and minuses of are those traditional methods as well. You're not going to get everything on a settle plate. It's just, it's so dependent on your airflow and dependent on movements. And again, probably thinking a lot about RABs and isolators. Your air samplers, you need another thing that I suppose is probably a pet peeve of mine, is really thinking about your flow rate to get that sweet spot. So it's not about how long it's going to take, it's getting the flow rate right so that you get that sweet spot of maximum capture and minimum destruction of cells, because they're already stressed. So there's a lot more to it, I think, than sometimes the less nerdy microbiologists along of us probably think about. But it's not just one answer. You need to really think about what your whole pattern of your EM is actually trying to achieve. I'm
And
thinking I
Kate,
may be on the same lines as you, Michael. I'm see you
you,
nodding
yeah,
your
yeah,
head there.
you certainly are. I've found so many companies that have not performed an appropriate risk assessment to
Yes, me too.
identify EM locations for different types of
Exactly.
monitoring devices, whether it's active air or passive settle plates or
Yep,
Rodax or swabs. And in the absence of doing that, it's not possible for you to have a robust environmental monitoring program
exactly.
because you
Yep,
haven't really identified the critical points of contamination. What's being is the process being contaminated? Is the product being contaminated? Are there open versus closed environments? In an isolator or a RABS, if you do have an intervention and you do have to open something up, where are the most critical locations? And what I found for companies who don't do that type of risk assessment for environmental monitoring sampling locations, then they've already lost the game before they started manufacturing.
I completely agree. And another one is even thinking and having good microbiologists that are involved in that process who are really thinking about what is the airstream that that microbe will be traveling along. Where are you actually intervening with your gloves? So if you've got your monitoring above that location and it's a settle plate, it's doing nothing because it's just your grade A airflow that it's from your UDAF. That
Yeah.
is all it's looking at. It's not monitoring any of your activities. So it's looking at where the glass line is, making sure that your locations are at or below that, so that you're actually capturing the actual areas where
Mhm.
the contamination is likely to reside from the activity. And I
Yeah.
agree. I think that's something
Mhm.
that is maybe not as robust
Mhm.
across our industry and the understanding of how to piece that together, or we don't give as much time as we should to that. But
Mhm.
yeah,
Sure.
definitely I'm very much aligned.
Yeah. Ian, I
Sorry, we've
think we
spent a
have two
long
more
time on that
questions.
question. Yeah,
Yeah,
yeah, it's an
Yeah, we
interesting
do.
one.
we did. It looks like we have a few more questions, which I'd love to get to because they're very relevant. So,
We'll,
Ian,
we'll
could you bring up
race
the next one?
into we'll race into the next one then. So, what
Yeah.
indicators would suggest loss of process capability in environmental control?
I'll let you
Well,
take this one first.
yeah, I mean, that's a loaded question. I mean, let's
It
define
is.
process capability. Let's define process capability. I think we've touched upon this earlier, and certainly in answering the last question as well, is there are a lot of things that need to be put in place even before you do your EMPQ or your routine environmental monitoring. And that comes back to the control of the environment, the control of people and material flow, the cleaning and sanitization programs. If you have an isolator or even a closed RABs, the type of gaseous validation that you're using to decontaminate those environments, maintenance of HEPA filters, your program for interventions and moving material from one place to the next, or even rapid transfer port opening and closing. Any one of those, and that's just a small, introduction, but any one of those activities and process controls, if one of them fail, then there could be a cascading event of having, complete loss of contamination control as well. So all of those pieces need to be in place. Those could be indicators, and indicators could be airflow, right? It could be information from the non-viable or total particulate continuous monitoring that you have as well, which is always going to be in real time. Any of those types of indicators or loss of contamination control obviously can play a role in an environmental monitoring failure. The interesting thing here is, unfortunately, there's no one-to-one correlation between loss of control in all of those areas and saying, oh, I'm always going to get an environmental monitoring hit, because historically speaking, we're only taking a very quick snapshot in time with an EM, if you're using a traditional
Yeah.
method. And you can never know that's where the beauty of these rapid continuous monitoring devices is that if we do have a loss of control, we have a better opportunity to see that loss of control, because now we're monitoring continuously, where historically it's been maybe once a shift or whatever it may be. Even with changing out a settle plate in one particular area every 4 hours, it's still not giving you a holistic view of control, just looking at environmental monitoring. So,
And
Kate.
I probably take this from the other side as well, that if environmental monitoring was seen as the only indicator of loss of environmental control, then Annex 1 would not have been revised to include all of the information on the contamination control strategy. So exactly what you're saying there, Michael, taking it from the other side, it's not one thing. And you need to make sure that your contamination control strategy implementation is robust, so that all of those iterative controls are, in place, that they're utilized, that they're compliant, and then you don't really need to worry about your environmental monitoring, and you've got a better chance than if you do have a signal, or one signal, and then you don't have a trend of being able to explain the entire holistic process. Whereas I think as an industry, we're still in that position of thinking that the one EM hit is the be all and end all, and we forget about everything else. Whereas actually Annex 1 has been written for the purpose of that is not your only piece of data that you use to define your environmental control. Obviously, if everything is sending signals everywhere and everything is contaminated, I'll eat my words. But when you're talking about that one hit, it's looking at everything together.
Yeah.
Excellent. So probably the last question we've got time to answer. Please clarify on which standards are followed in undertaking the validation and comparability assessment. What would a typical workflow for this look like? And is the criteria purely statistical?
Right. So generally speaking, when we're looking at, I wouldn't call them standards, but these are guidances on the validation of alternative and rapid methods. There are three areas that we can look to. One of them is European Pharmacopoeia Chapter 516. Another is the United States Pharmacopoeia Chapter 1223. And of course, the PDA Technical Report number 33. The two compendia chapters are fairly general in providing guidance on how to demonstrate comparability for qualitative and quantitative methods. They don't do a really deep dive into how to select the right statistical analysis. There is some mention, but how to actually do this based on the data sets, or even going into some of the systems we've talked about today, including continuous monitoring systems, and how do you make those comparisons. So because of that, I would refer our listeners to the TR33. Again, in the next couple of weeks, this will be fully published. To drill down to the second part of the question, what is the typical workflow and what's the criteria? Is it purely statistical or not? It certainly is a combination. So when we look at, for a quantitative method, for example, we would normally look at the ability of the alternative method to show comparability to the existing method in terms of accuracy, repeatability or intermediate precision, linearity, range, and limit of quantitation. How low can you go and get a numerical value with good accuracy and precision? And for a lot of rapid or alternative methods, the end user can actually perform these and use the statistical recommendations that we've put out in the TR33. We now have an annex, which is almost as large as the main technical report on how to do statistics, which the industry's been asking for the last 15 years. So we've put this in place. But for continuous monitoring, environmental monitoring devices such as the biofluorescent particle counters, it's not possible for the end user to challenge the system. Okay, if you try to put a solution of microorganisms into the systems, you're going to burn the optics, and then you're going to end up with a paperweight, right? So in those instances, the only thing you can do is the side-by-side comparability testing in the appropriate areas. So where do you rely on the data for accuracy and limit of quantitation and so forth? You go back to the primary validation that the supplier is expected to have performed. And most of these suppliers have the capability of challenging with aerosol standardized suspensions in controlled environments to do those side-by-side tests. So I would tell the listeners that information, go back to the suppliers and request the primary validation data that they have. If they don't have that information, you may want to look at another supplier, okay? Because that is an expectation of all suppliers developing these methods. So hopefully we've been able to provide the listeners, Ian, with good direction on where to go for all of this information.
TR33 is the one to remember for anybody that needs the reminder. And
TR33, yes.
yeah, and we
Yeah.
better leave it there, Ian. I know everybody will be trying to get back to work. So
Yes,
thank you very much
absolutely.
to everyone for listening.
And thank you very much, Michael and Kate. You've been a fantastic duo. I have really learned a lot, including how to pronounce Rhodout, which I didn't know we ever do before. Thank
Fantastic.
you both. Thank you to our sponsors, Thermo Fisher, without whom none of this could happen. So please do support them as much as you can. So our next session, which starts in about half an hour, well, actually 25 minutes, is on one of those topics that we cover off regularly. And it's a topic that I think everybody would wish us to not be able to cover because it's gone away. It's nitrosamines, which is always going to be a hot topic for us. If you'd like to join us for that, that would be fantastic. But with that, once again, thank you both. And let's bring this session to a close. Thank you all.
Thank you.
Thank you.